Hi everyone. Today we're going to be looking at a study from Ferrari Sousa and colleagues, published in Nature Neuroscience. They're investigating the critical interplay between microglial activation, amyloid-β pathology, and astrocyte reactivity in the context of Alzheimer's disease.
All right, so the study kicks off by addressing a well-known gap in Alzheimer's research. For a long time, the main characters in the story have been amyloid-β plaques and tau tangles. But the thing is, these two proteinopathies, they just don't fully explain the clinical progression of the disease. It's clear that other mechanisms have to be involved.
And that observation naturally leads us to the question of neuroinflammation. Specifically, what's the role of the brain's resident immune cells, the microglia and the astrocytes? The big question this study tackles is whether the activity of these cells is the missing link in that pathological chain. So, with that in mind, the authors put forward a very specific hypothesis. They focused on the crosstalk between these glial cells. Their idea was that microglial activation acts as a key modulator. In other words, it's an essential preceding step that's required for amyloid-β pathology to trigger that reactive state in astrocytes, a process we call astrogliosis. Okay, so how do they test this? Well, the researchers used what's called a multimodal biomarker approach. This means they combined advanced imaging with fluid biomarker measurements, and they did this in two separate cohorts. This design is quite robust because it allows for a primary analysis and then a chance to replicate their findings. Let's look at their primary sample. It was made up of 101 individuals from the triad cohort. And to make sure they were capturing the full clinical picture, this group included both 62 cognitively unimpaired people and 39 who were impaired. The mean age was about 72, and around 35% were carriers of the APOE4 allele, which we know is a major genetic risk factor. Now, for the biomarkers themselves. To measure microglial activation, they used PET imaging with a specific radiotracer, 11C PBR28. This tracer is useful because it binds to a protein called TSPO. And since TSPO is upregulated in activated microglia, the PET signal gives them a quantitative in vivo measurement of this specific cellular state. And what about the astrocytes? For their primary outcome, astrocyte reactivity, they measured plasma levels of glial fibrillary acidic protein, or GFAP. GFAP is an intermediate filament protein that gets released by astrocytes when they become reactive. This makes it a very reliable and, importantly, an accessible fluid biomarker for tracking astrogliosis. Of course, they needed to measure the other core pathologies as well. They used specific PET tracers to quantify A-beta plaque deposition and aggregated tau tangles. They also measured soluble phosphorylated tau in the plasma. And one more important point, they also used soluble TREM2 from cerebrospinal fluid. This served as a secondary marker for microglial activation, which was especially useful for their larger replication cohort of 251 people. All right, so with that background on the study design, let's dive into the primary findings. This is where they really start to test their core hypothesis about the interaction between A-beta, microglia, and astrocytes. The regression analysis here revealed something quite striking. When they looked at individuals who were negative for microglial activation, there was no significant link between their A-beta burden and their levels of plasma GFAP. But in the group that was positive for microglial activation, the story completely changed. A higher A-beta burden was strongly and significantly associated with increased astrocyte reactivity. And this wasn't just some artifact of splitting people into positive or negative groups. When they treated both A-beta load and microglial activation as continuous variables, the statistical interaction between them was also highly significant. This really confirms a synergistic relationship. It suggests the effect of amyloid on astrocytes is progressively amplified as microglial activation increases. And remember that replication cohort? Well, they successfully replicated these results in that larger sample of 251 people. This time they used CSF STRUM2 as the marker for microglial activation. As you can see from the data, the magnitude of the association between A-beta and GFAP is much larger in the STRUM2 positive group. This confirms that modulatory role of microglial, but now using a completely different biomarker, which adds a lot of weight to the finding. So the next logical step was to look at the downstream consequences of this glial crosstalk. The crucial question becomes, does this interaction actually connect to tau pathology? After all, tau is the feature of Alzheimer's that correlates most closely with neuronal death and cognitive decline. To get at that question, the investigators checked to see if this AB-driven astragaliosis, the one that happens only when microglia are activated, was associated with tau. And they looked at both forms, the early soluble phosphorylated tau and the later aggregated tau tangles that you can see with PET imaging. And what they found was a pattern that was identical to what we saw before. Higher astrocyte reactivity, that is, higher plasma GFAP, was significantly linked to higher levels of soluble phosphorylated tau. But again, this was only true in the individuals who were positive for microglial activation. Without that microglial activation, the relationship disappeared. Disconnection wasn't just limited to soluble tau either. It extended all the way to the later stage of insoluble, aggregated tau tangles. They found a significant association between astrocyte reactivity and the tau PET signal in neocortical regions. And, you guessed it, this was exclusively within the microglial activation positive group. This is a key point, as it suggests this glial interplay is linked to the entire spectrum of tau pathology. Okay, so for the final piece of the puzzle, the researchers used a technique called structural equation modeling. This basically lets them take all these different biomarker findings and integrate them into one comprehensive model. It's a way to statistically test how this proposed biological cascade connects all the way to the ultimate clinical outcome, which is cognitive impairment. Let's start with the group that lacked significant microglial activation. In these individuals, the proposed cascade model just didn't work. It was inadequate. Yes, amyloid showed some direct effects on tau, but there was no significant pathway linking any of these biomarkers to cognitive impairment. On top of that, the model itself was a poor statistical fit to the data, which we can see from the high RMSEA and low CFI values. But now, let's look at the microglial activation positive group. Here, a very different picture emerged, a coherent and statistically robust pathological cascade. It goes like this. A-beta load influences astrocyte reactivity. That astrocyte reactivity then partially mediates amyloid's effect on tau phosphorylation. Higher phosphorylated tau in turn is associated with more tau tangles, and finally, those tau tangles are directly linked to cognitive impairment. And this wasn't just a weak association. This model was highly explanatory. The R-squared value was 0.76, meaning this exact sequence of events accounted for 76% of the variance in cognitive scores in this group. The statistical fit was also excellent. An RMSEA of 0 and a CFI of 1 essentially signifies a perfect match between their proposed model and the data they observed. So to summarize, what this study provides is significant in vivo evidence from humans for a very specific sequence in the Alzheimer's pathological cascade. It's a sequence where glial cells aren't just bystanders. They play a central modulatory role that seems necessary to bridge the gap between the core pathologies and the clinical symptoms. We can break down the key conclusions into 4 main points. First, microglial activation seems to be a prerequisite for amyloid-β to trigger significant astrocyte reactivity. Second, this glial interaction is then directly linked to the downstream development of tau pathology. Third, this entire cascade from AB through glia to tau is strongly associated with cognitive decline. And finally, this all points to glial cells as potentially important therapeutic targets for modifying the course of the disease. Now, the authors are careful to acknowledge several limitations, which is standard for this kind of research. For one, the study is cross-sectional, which means it establishes strong associations but can't definitively prove causation. Also, while the biomarkers are state-of-the-art, they measure general glial reactivity and can't capture the full complexity of different glial phenotypes, some of which might be protective. And of course, using volunteer cohorts always carries a potential for some self-selection bias. To wrap up, the paper concludes by reinforcing this powerful pathological model. The core idea here is that neuroinflammation isn't just a secondary reactive phenomenon. Instead, it seems to be a core mechanistic hub, one that links the classic proteinopathies of amyloid and tau to the actual clinical manifestation of cognitive decline. This provides a much more complete and I think more accurate framework for understanding how Alzheimer's disease progresses.